How does an insect climb straight up walls or windows without losing its grip? And how does a snake slither across sharp rocks without slicing itself open? Can engineers use this knowledge to improve biomedical technology? Stay tuned for some sticky and slippery science. From the College of Engineering at Oregon State University, this is Engineering Out Loud. I'm Keith Hautala. This season, we're focusing on research that keeps us healthy and safe. I'm going to take you on a journey that will follow the tiny wet footprints of a ladybug, and we'll go inside the mouth of a frog. But it all starts with the secret language of lobsters.
So actually, my very first project in this field was when I was just finished undergrad, and I started working in a lab doing biomechanics work. We're looking at fast animal movements. Lobsters actually make noise. They talk to each other. They make these little scratching noises.
That's Joe Baio. He's an assistant professor of bioengineering here at Oregon State.
And if you look at that mechanism, it's a stick and slip. So they have this little violin bow on their antennae. And it sticks, and then it slips, and that's what makes a little noise, like a scratching noise. And so we were looking at that stick and slip mechanism, and then that kind of got me in that's what's going on with a slug or a snail. It sticks and then slips, and that's how it kind of slides.
If you've ever sat and watched a snail or slug moving, it is kind of fascinating. That trail of slime it leaves behind actually plays a key role in how it gets around.
There's something about the chemistry there that's interesting, right? It for some reason can, under certain environmental conditions, get sticky and provide a lot of friction for the animal movement. And then all of a sudden it turns off and allows the animal to move freely. So it becomes slippery. So that's kind of how I got into the adhesion business, or the interest in that. And then you start looking at all kinds of animals, right? They can stick to all kinds of things.
If you were going to try to come up with the first law of sticky science, it might be that for everything that wants to stick to something, there's something else that doesn't want things sticking to it.
And then on the opposite end, there's all kinds of plants, right? So plants don't want insects walking on their leaves. So they come up with crazy waxy surfaces that prevent insects from walking on them, prevent insects from eating them and destroying the plant. And so there's just tons of different systems out there that rely on these dynamics between sticky and slippery. And we're just starting to look at them at the molecular level.
These molecular mechanisms evolved over very long periods of time, basically through trial and error.
Yeah, so if you look at the natural world, there's all these extreme properties. There's materials, whether it's skin or a plant leaf, that really just have adapted to these really extreme temperatures, or to deter predators, or to be able to stick to something, or to be able to prevent their skin from falling off when they slide on a rocky surface or something like this. And evolution has gone through thousands and hundreds of millions of years to find solutions to these problems. And we engineers are just starting to kind of look at them and look in the natural world for solutions to some of our problems.
There are all sorts of applications in medicine, particularly for getting things to stick where you want them to and to not stick where you don't want them to.
Trying to come up with coatings that maybe don't wear down, like a coating that you could put on a artificial hip so that it doesn't wear down, or that it prevents bacteria from adhering to it, or a new type of adhesive that can basically turn on and off. Can we get away from stitches, right? Or staples, and glue tissue together, right? Or glue dental implants in, and maybe do it in a way that's biocompatible, right? We're using kind of biological molecules, so maybe it's less likely to kind of cause a problem in your body when you put it in. And also looking at these adhesives from nature is that they turn on and off.
That's kind of a key selling feature of these natural adhesives, the ability to turn the stickiness on and off. Imagine a bandage that would stick to your elbow all day without falling off, but then it peels off easily without ripping off 2 layers of skin.
So let's say a fly or some spiders even, or ladybugs is what we actually study in our lab. They have these little hairy feet, and they look similar to a gecko foot, where they're full of hairs. And unlike the gecko, these insects also have a little fluid. So these hairy feet bend, and they make a really nice contact to whatever substrate they're trying to adhere to. And then this fluid also gets, basically emitted from their feet and kind of also helps make contact. And kind of the theory was that this fluid can adapt, right? So a spider or a ladybug.
So This mysterious ladybug foot juice changes properties depending on what kind of surface the ladybug is walking on. If that sounds like there's some complicated chemistry going on, well, there is.
So super complicated. It's like an emulsion of proteins, like all kinds of biomolecules. So lipids, so these fatty molecules, proteins, sugars, water, other acids. And we were thinking maybe like different parts of these things are important depending on what substrate they're walking on, right? Evolution has over-designed it. And so we were looking at, okay, let's let an animal walk on all kinds of different substrates and look how this fluid actually adapts, and adapts at the chemical level.
When it comes to sticking and slipping, all of the important action takes place where the rubber hits the road, so to speak, at the surface.
We are kind of surface scientists in my group, and so we're really interested in what's going on at the outer few molecules of an interface, right? Whether it's the surface of a silicon wafer and a computer chip or the surface of a ladybug's foot.
To figure out what happens on the surface of a ladybug's foot, Joe and his colleagues created a whole bunch of designer surfaces with different properties for the ladybugs to walk on.
And when the ladybug walks on it, it leaves actually a little wet footprint. And then we immediately take that footprint and do our chemical analyses on it. We kind of look through what are the chemical components that actually interact with that substrate. And then we maybe look at how that changes as it walks on different substrates. And so then we can look at the physics and the mechanism of how different parts of this fluid come down and bind to a substrate versus maybe bind to the foot edge of the ladybug. And then we can take all of what we know about these components, maybe put something that's similar in a beaker, and then make our own sticky ladybug fluid. And we've been mildly successful. We're at the stage now where we know what's going on, kind of like the different chemistries and what's actually interacting, and we're still in the process of making a mimic and then applying that mimic to biomedical applications.
Another sticky mystery concerns the tongues of frogs. When a frog sees something it wants to eat, say a nice tasty cricket, it shoots out its tongue. The tongue sticks to the cricket, and the frog pulls it back in.
So something about the frog tongue chemistry makes it sticky, but it can't be sticky when it's in their mouth, right? They'll just stick to the roof of their mouth. So when a frog hits a prey item, right? So the frog extends its tongue, it has a bunch of this fluid on its edge of its tongue, hits a fly or something it wants to eat, it sticks to it and retracts it back in. So there's some sort of chemical change going on, or all of a sudden, how does this become sticky as it hits the fly surface? And so we were interested in looking at that mechanism too. And so my graduate student made a bunch of substrates with different chemistries on it and allowed a frog to hit it, right? We basically these little substrates were clear, and we stick a fly behind it and trick the frog into whacking our sample with its tongue.
So what they end up with is a frog tongue print on a glass slide.
And what's left there is the spit of a frog. And if you look at the chemical structure there, you notice that it's different when it's on its tongue versus at the surface. So when it's at the surface, as it sticks, there's a bunch of fluid between the tongue and the substrate. As the tongue is retracted, it again applies this kind of force, this kind of shear force. And then that causes basically the molecular structure to change. It almost causes these little molecules to form fibrils, almost like a rope, and it gets really, really strong and sticky. And that allows the frog to retract its prey. And so again, this idea of on and off, right? By applying a certain force, you can turn the stickiness on or off or adjust it in different ways. And what's really interesting to me is just this crazy idea that force, like something physical, can change the chemical structure of these kind of fluids.
In the frog's saliva is a kind of mucus which contains these big molecules called mucins. These are basically just proteins with lots of sugars stuck on them. But what Joe and his collaborators found out is that in their off state, these mucins are just kind of a disordered mess pointing in all directions. But when they stick to a surface and get stretched, they interact with each other. They start to stack up and form these fibrils that are very strong and very hard. And nobody really knew how this worked before. It took some really smart bioengineers to figure it out.
And the way we kind of did this is we do a lot of surface analytical work. So again, going back to my student, we had this frog print, right? So then we have this goo on a slide or on a substrate, and then we take it to the synchrotrons. And so this is where, a synchrotron, we're using tunable X-rays. So they spin electrons around very fast and create tunable X-rays that we use. So we hit this surface. We have our surface with our spit on it. We hit it with X-rays, and what comes off are other light photons, so pieces of light. And then we measure the kinetic energy of this light that comes off, and that can tell us what's at the surface, what molecules are there. And the cool thing is it's also sensitive to order. So the way we set up the experiment is we change the way the light interacts, so the polarization, the way the light hits the surface in certain angles, and we look for changes in the response. And that can tell us, oh, all of these molecules are well ordered or all of them are disordered. And so that's how we could tell that this fluid turns on after it hit the surface, right? Hits the surface, it undergoes an ordering process. All these molecules order in a very distinct way, and we were able to see that with our spectroscopy.
So the frog tongue mucus is basically a pressure-sensitive adhesive that turns on whenever force is applied to it.
It's super fast, right? It becomes sticky almost immediately. It has to, right? Or else the prey item can just disappear or fly away or escape. And so that's, I think the next step is understanding the kinetics. So how fast is this kind of chemical reformation process, right? How it can go from disordered to ordered that quickly is super impressive.
This kind of pressure-sensitive adhesive could find some novel, if not downright revolutionary applications in biomedical fields.
Yeah, again, gluing really crazy tissues together, right? During surgery. So let's say you cut open a heart and you need a glue that works really fast, right? To prevent the patient from bleeding out. Or something that, you just want a temporary adhesion to something, right? You want to be able to hold pieces of tissue together and then be able to reopen them, maybe in later surgery, these sorts of things. Or just a way, you don't have to put a lot of foreign things in your body. These are proteins. These are fat molecules, maybe some water and a little bit of fatty acids and things like this. And so the body might not see it the same way as sees like a huge piece of metal staple, right?
So ladybug feet and frog tongues are just two examples of completely different mechanisms on the sticky side of things. On the other end of the spectrum, we get into the slippery stuff. Things like snakes. What sort of useful tricks can they teach us?
The one example I give is the ball of an artificial hip, right? It's rubbing against the other part, the cup of your hip or the artificial cup, and over time wears down, right? And so then you have to replace that hip. And so could you come up with a way to make it slippery, right? So you prevent this abrasive kind of process. And, I was talking to a colleague, and he's like, snake skin has to deal with this all the time. We studied the king snake from California, and they're on all kinds of like crazy sharp rocks. And while they do shed their skin, it's pretty anti-abrasive, their scales. And so they can slide over and over on something that's really sharp or really rough, and their skin maintains its integrity. And it's also self-cleaning, which is a whole other thing, is just fouling, so it prevents stuff from adhering to it. If you can create something that's self-cleaning or non-fouling, that you're gonna make $1 billion. Catheters you stick into a body, if you make them non-fouling, bacteria can't stick to them. Or a stent, when you stick in a stent in your bloodstream, preventing blood clots and things like that. So this idea of having something really anti-abrasive and non-fouling or self-cleaning is super impressive, this material.
Snakes are a lot more slippery on their bellies than they are on their backs. Joe and his colleagues figured there was some kind of specially adapted biomolecular mechanism at work.
Again, we took these samples and kind of chemically imaged them with our surface analytical tools and found that on the belly of the snake, where it's slipperier, there are these basically fat molecules, kind of like these really long chain carbon molecules, and they form a really beautiful monolayer at the snakeskin surface. And if you look at the top of the snake, where there's higher friction forces, they have these same molecules. The way they're interacting with the surface, the way they're pointing, the way they're ordered is way different. So on the bottom, it's well ordered, well stacked together, like all lined up. And then on the top, they're kind of disordered again. And so this well-packed layer of oil basically allows these snakes to slide on a substrate or a surface without a lot of abrasion. And then this layer also provides, I think, some non-fouling capabilities. And so we've started to identify what molecules are actually at these surfaces, and then trying to come up with a way to easily coat like a biological implant, an artificial hip or these sorts of things. Is there an easy way to create a coating that exactly mimics the chemistry at like a king snake skin surface?
Joe says he gets a lot of inspiration from the natural world, whether he's reading a biological science paper about it or just out living in it, taking hikes with his young son or exploring his own backyard. And it's something he says all kinds of engineers can benefit from.
There's all this research also that just being out in nature, really calms your nerves and inspires you. And I think just being able to look at biology. I think more engineers need to look at biology instead of just looking at what other engineers have done, right? So when most engineers take a product, where they're designing a new engine for a plane or a new type of diabetic glucose sensor, look at what's previously been done and kind of make iterations on that. But I think this idea of kind of going back and looking at what these problems nature's already solved and how they do it, and can you kind of take any inspiration from that is key. I think by studying nature, we can make some really cool discoveries that can have a huge impact in medical field or in all kinds of different fields.
This episode was produced by me, Keith Hautala, with additional audio help from Molly Ayton and Rachel Robertson. Our intro music is The Ether Bunny by Eyes Closed Audio on SoundCloud, used with permission under a Creative Commons Attribution license. Other music and sound effects were also used with appropriate licenses. You can find the details in our show notes, which are online, along with other episodes at engineeringoutloud.oregonstate.edu. Subscribe on our website or by searching for Engineering Out Loud on your favorite podcast app. Don't you want to glue bloody tissues together? I mean, I guess that would be the ultimate thing for the, first aid kit, right? Like, glues bloody tissues together. Like, oh, God, where's the goo? It would be cool.
This needs to go in the podcast.
No.
That's the ending. After everything already sounds like it's
the outtake reel.
First aid kit.
Okay.